Hi friends, this is Vasa Subbarao along with Manu welcomes you to Green Coach. Green Coach offers online tutorial for various horticulture subjects. In our today's class, we are discussing about entomology and we are discussing about the microbial method of pest control. Let's start our today's class. The microbial method of pest control. Using microorganisms for the control of pests is called microbial method of pest control. Uses of microorganisms for the control of pests can be termed as microbial method of pest control. It was discovered by the contribution of A.M. Bassi. Microbial method of pest control was discovered by the contribution of A.M. Bassi. In the year of 1835, he observed the white muscadine disease in silkworm. In the year of 1835, he observed the white muscadine disease of silkworm. The white muscadine disease of silkworm is due to the fungus called Bavaria bassiana. By observing this, he is termed as the father of insect pathology. The father of insect pathology is Agustina Mario Bassi. What is the advantage of microbial control? Means the insect does not develop resistance quickly to the microbial controlling measures or the microbial insecticides quickly. The insect do not develop resistance to the microbial insecticides quickly. In the microbial method of pest control, mostly the bacterial pathogens are used. Bacillus thuringiensis species are mostly employed. The bacillus species are identified by Ishivata. The bacillus species are isolated by Berlins in Thuringia in Germany. Hence these are called Bacillus thuringiensis. The species name thuringiensis is named after Thuringia and Bacillus is named after Agustina Mario Bassi. Bacillus thuringiensis produce two types of toxins. They are exotoxins and endotoxins. Bt toxins are of two types, exotoxins and endotoxins. The exotoxins are of two types, alpha exotoxins and beta exotoxins. And the endotoxins are delta endotoxins. Alpha and beta endotoxins are non-specific and show minor effect. Alpha and beta exotoxins are non-specific and show minor effect, but the delta endotoxins are specific to host and show major effect. Let's see the mode of action. Here I'll make it a flow chart for your easy understanding. The Bacillus thuringiensis or the bacteria enters into the body of pest through mouth. It reaches the midgut and the protoxins are formed. These protoxins are in inactive form due to the alkaline pH of gut, that is 8 to 10.5. The alkaline pH of gut should range between 8 to 10.5. In this pH, the protoxins are converted into endotoxins. They bind to the gut receptors. Which leads to cell lysis or gut paralysis or septicemia. Which ultimately leads to the death of insect. I'm repeating the flowchart once again for you friends. The bacteria enters the mouth of pest and from the mouth it enters the midgut and forms the protoxins which are in inactive form due to the alkaline pH of gut that ranges between 8 to 10.5. They become the endotoxins which bind to the gut receptors, which leads to cell lysis, gut paralysis or septicemia and ultimately death. What do you mean by septicemia? The whole hemolymph or hemocoel of insect is filled by bacteria. Which deprives the insect to nutrients. Ultimately it causes death. Septicemia is the condition where the hemocoel of insect is fully covered by the bacteria which deprives the insect out of nutrients. It ultimately causes the death. Synergism. Synergism. Synergism is the principle where the Bt toxin's toxicity is increased. The Bt toxin's toxicity is increased by the addition of carbaryl. Synergism is the property due to which the toxicity of Bt cotton is increased by the addition of carbaryl. Let's see about the toxins to the each order. Cry1 toxins are used for Lepidoptera. Cry1 toxins are used for Lepidoptera. Cry2 toxins are used for Lepidoptera plus Coleoptera. Cry2 toxins are used for Lepidoptera and Coleoptera. Cry3 are used for Coleoptera. Cry3 are used for Coleoptera. Cry4 used for Diptera. Cry4 used for Diptera. Cry5 used for Lepidoptera and Coleoptera. Cry5 used for Lepidoptera and Coleoptera. Cry6 used for nematodes. Cry6 used for nematodes. This step is very important. Cry1 for Lepidoptera, Cry2 for Lepidoptera and Coleoptera, Cry3 for Coleoptera, Cry4 for Diptera, Cry5 for Lepidoptera and Coleoptera, and Cry6 for nematodes. This Bt cotton method has several disadvantages also. Though it is the complex mechanism, some insects develop resistance to Bt. These are at field level and at greenhouse level. At field level, diamondback moth, which is termed as Plutella zytostella. Plutella zytostella of crucifix developed resistance to Bt varieties. Diamondback moth, Plutella zytostella developed resistance to Bt varieties. At greenhouse level, cabbage looper, Trichoplusia also developed resistance. At greenhouse level, cabbage looper, Trichoplusia also developed resistance. Let's see the summary, friends. Microbial method of pest control. Mostly bacillus species are employed because of their Bt toxins. There are of two types, exotoxins and endotoxins. Delta endotoxins are most effective and host specific. And here the flowchart. Bacteria enters the mouth and it reaches the midgut of pest. It releases the protoxins. Due to the alkaline pH of midgut, which is pH ranges between 8 to 10.5, they becomes endotoxins. Binds to the gut receptors, leads to cell lysis or gut paralysis or septicemia, and ultimately the death. That's all for now, friends. Thanks for watching Green Coach videos. Please like and subscribe our channel for further video updates. We will update you soon with another video. If you want to give us any suggestions, please comment them in the comment section. Keep supporting us and thanks for your love and support. Thank you guys. This is Subbarao along with my friend Banu signing off for today. Keep supporting.